Welcome back to Plantopia. Plantopia is the plant health podcast of the American Phytopathological Society, and I'm your host, Matt Kasson. I'm an associate professor of forest pathology and mycology at West Virginia University. The views and opinions expressed by our guests are their own and do not necessarily reflect the views of the host, the American Phytopathological Society, or any affiliated entity. This is the second episode of 2026. Unlike previous Plantopia episodes, this episode includes 4 guests: Dr. Carrie Harmon from the University of Florida, Dr. Alicyn Smart from the University of Maine, Dr. Peng Tian from the University of Missouri, and Dr. Matt Bertone from NC State University. All 4 will join me to discuss their roles in plant disease and pest diagnostics as part of the National Plant Diagnostics Network, or NPDN. I'll first speak with Dr. Harmon about the NPDN, its history, and the role it plays in food and plant security across the United States, and then more specifically about plant diagnostics at the University of Florida. After that, I'll have a 4-way conversation with Alicyn, Peng, and Matt about their own roles in the NPDN, misconceptions about plant disease and pest diagnostics, and the many challenges they face. I hope you enjoy this new format, and we'll let Carrie introduce herself.
Hi, I'm Carrie Harmon. I'm a plant pathologist and the lab director at the University of Florida Plant Diagnostic Center. In that role, I also help to run the National Plant Diagnostic Network and the southern branch of it, the Southern Plant Diagnostic Network. But I'm a plant pathologist that's been doing diagnostics for a little over 20 years.
So you've been in the clinic for that long since you started at UF?
I started actually to help run the Southern Plant Diagnostic Network at the beginning of the NPDN. So the NPDN was created in 2002, but really got off the ground hiring the people to run it in 2003, which is when I joined the faculty here at the University of Florida to do that job. In 2009, I was asked to take on directorship of the Plant Diagnostic Center, the clinic.
And the NPDN, just to remind our listeners, consists of a network of more than 70 labs, 200 professionals, mostly affiliated with land-grant institutions across the United States.
That's correct. And to remind people what we mean when we say land-grant institution or LGU, land-grant university, these are usually the flagship agricultural universities. There's one in every state, sometimes more than one, but usually the big one in every state that is tasked with extending practical knowledge from research out to the population so that they can improve their lives. So the diagnostic labs are usually linked in through those institutions because it's a service of that institution to serve its clientele, who are the citizens of that state.
That makes sense. And there's 5 regional PDNs based on the kind of geography where they are in the United States. And you said you oversee the southern division of that, but there's also the Northeast and North Central, the Western, and was the Great Plains. Is that the...
That's right. And they look just based on geography, but it's actually based on cropping systems and agricultural inputs and outputs. So yes, it ends up being by geography, but it's really because of the agricultural and green industry trying to centralize the knowledge and connections between those diagnostic labs and professionals across those states because they're dealing with similar crops.
Now, as a plant pathologist, I can't help but think that the timing of the NPDN sort of lines up with Phytophthora ramorum. And I wondered if that was a big impetus for establishing the National Plant Diagnostic Network.
Actually, it's the other way around. The National Plant Diagnostic Network preceded both soybean rust and sudden oak death. It's a good thing that we were in place when those 2 diseases hit. So the National Plant Diagnostic Network was actually developed in response to 9/11, when we realized that not only could our buildings and infrastructure be at risk, but so could agriculture. So Congress, through a cooperative agreement through the USDA, said, figure out how to make sure that our crops and the way that we feed, clothe, and shelter humans in this country, make sure you can safeguard that. And the National Institute for Food and Agriculture developed, with collaboration from those land-grant universities at the 5 regions, figured out what we were going to do to network the diagnostic labs and also extension professionals for most of the crops that we grow. And that meant that we were on the ground and ready to receive samples when sudden oak death was first detected, but also when soybean rust was first detected. So not just trees and forested areas, but also our agricultural major breadbasket crops. So NPDN is the National Plant Diagnostic Network. We really process the samples to say, is this pathogen here or not?
Yeah.
But then what you do for collecting those samples before it gets to us, and then what happens with the information that comes from our diagnostic labs, whether the pathogen is detected and it needs to be managed or eradicated, or it's not detected and we need to figure out what the disease actually is. NPDN has been involved in the development and expansion of lots of programs like the one for sudden oak death or Phytophthora ramorum.
Yeah.
So that we can connect the green industry folks who are being impacted by the detection of new things to the people who can help figure out whether it's going to impact trade, how to minimize impact on their business, and then increase food and plant security.
That makes a lot of sense.
The tests that we're running in diagnostic labs, every single sample represents a person who needs an answer and has to do something about it to bring that product to market or to be able to plant it or to be able to manage it. Sometimes we can put a value, a dollar value on, say, a greenhouse fold lettuce or a single azalea. But it's really hard when you start talking about a sentimental thing like 100-year-old historic trees.
I think that there are some misconceptions in plant disease diagnostics. And one of my takes is that people don't understand that these tests cost money. Do you think that is a misconception?
For sure. Unless you work in the field, whether it's human diagnostics or plant diagnostics or anything else, you don't really know what it costs for all the back-end stuff that produces whatever it is that you're accessing. So the grower, if they've sent me a sample, if we have this hydroponically grown lettuce, they are worried about the entire system going down based on whatever I find in that plant. So the value to them that day might be the value of the whole crop.
Right.
On another day, the value of that head of lettuce might come from somebody who's growing it in their backyard. They grew it from seed, so how can it possibly cost $40 for me to tell them why that lettuce is wilting? It's because there are people who've been trained that take decades of training. So I've been doing this for more than 20 years.
Mhm.
My grad students have been doing it for 4 or 5. These people have been trained to recognize many different diseases across many different hosts. So you're paying for that expertise, just like you pay at the doctor's office. It's also the technology that goes into bringing it to you. So we don't do everything just by a microscope, and I can't do everything just by looking at the sample. Sometimes it can't be cultured. So I have to look for the DNA or the RNA of these things, and that requires some really high tech. The more specific the diagnosis needs to be, or the more valuable the crop is to the person submitting it, the more high-tech we can go in the sample diagnosis if needed. So every single diagnosis, we're trying to decide what tests are most appropriate for that set of symptoms on that plant, but also where it's coming from. They've got to know exactly what the organism is so that they know exactly how to manage it. The reason it's so critical for us to know exactly what it is the management tools that we have at our disposal these days are extremely safe and very targeted. So with plants, we have a limited vocabulary to describe what the problem is. It died, or the leaves died, or it wilted, or it turned yellow. Those are all like the cough and the runny nose in humans. They don't say much except we need to ask more questions. Diagnostics is what allows us to ask those questions of a plant when it can't talk. We're able to open up the plant in a way that we can't really open up a person and test for based on our experience. And what I teach all my students is we always go and look for horses. When you hear clip, clop, you look for the horse, not the zebra.
Right.
Now, the diagnostic network is set up to look for both. Our job is to learn what all the horses are and to be able to recognize those horses and recognize when it's not a horse. Not necessarily that it's a zebra, just that it's not a horse. And then we can call in specialists who can help us identify that it's a zebra. So I think I don't know if I'd call it a misconception, but I think it's just a lack of awareness that what it takes to identify the source of a problem requires a lot of information from both the sample submitter, stuff about the environment, stuff I have to learn what the plant is. Knowing those things really helps me explain to my clientele why it takes as long as it does, 3 to 5, even 7 days to turn around a diagnosis, why it costs what it does, what the tests are that I'm offering, what the limits are of those tests, and then what my answer is not just the diagnosis, it's disease X, but also what they can do about it.
What's the craziest thing you've seen or experienced in the clinic?
There are a couple. One that lots of diagnosticians will probably mention. That's delusionary parasitosis, and it's actually taken very seriously. But as far as like plant samples that I've had that are totally crazy, I would say rather than it being something funky I diagnosed, it would be the sheer volume. One year, there was a new virus in town across the United States. It happened to hit right before the winter holiday break. And my lab received more than 20,000 samples. We turned my classroom and laboratory into a giant assembly line of diagnostics where 2 people were taking the samples out and giving them numbers and making a record of them. 2 people then did the next thing, which was to grind them in the special bag. The next person took out some of the liquid and put it in the special buffer and that kind of thing. And then the next person put a timer on them and watched for the result and recorded the result. And then the last person was doing sort of the cleanup and make sure that we kept all the stuff contained, organized, and recorded. That was the craziest. I have never had that kind of a surge. We had to basically stop doing everything else. I still don't know if it was a good thing or a bad thing that happened right as we were going into break. And maybe a good thing because people didn't have classes, and so I had all hands on deck, and we could say no to all the rest of the samples.
What was the virus?
We were running a test for the parent virus, which is tobacco mosaic virus.
Okay, I thought it might have been plum pox or something like that, but
Nope, there are protocols for that too when we get surges like that. And so we were getting seedlings, little tiny bits of plant material, teeny tiny bits of plant material, but we were getting in hundreds from single greenhouses all across the country. And we had to keep them all straight. And then we had to report out very quickly because in a case like this, and this is a perfect example of why the network is in place, to deal with surge like that. And we happened to be one of the few labs that was available to do that right then. But the coolest part of it was that we were able to pull it off. And we were then able to transfer that protocol. We actually ended up testing a protocol, identifying a problem with the protocol, switching to a different method, and backing up APHIS so that we can then make recommendations to other labs.
sounds like a functional network to me.
We can laugh about it now, but that would be my craziest diagnostic story, I think, is processing 22,000 samples in a week and a half.
Now that Carrie has given us an in-depth understanding of the NPDN and its history and disease diagnostics, I want to bring in our other guests to share their own perspectives on these same topics. I'll let them introduce themselves.
I'm Alicyn Smart. I'm Associate Extension Professor and Director of the University of Maine Plant Disease Diagnostic Lab, and I've been here 9 years.
Nice.
My name is Peng Tian. I'm an Assistant Extension Professor at the University of Missouri. I'm also the Director of MU Plant Diagnostic Clinic. February this month is actually my 5-year anniversary working with Mizzou.
Welcome.
Hi, my name is Matt Bertone. I am the entomologist and director at the Plant Disease and Insect Clinic at NC State University, and I have been in the role for almost 13 years now in March.
Okay, great. Well, it's nice having all three of you on. I'm excited to talk to you. When I was talking to Carrie earlier, we were talking about her role as part of the National Plant Diagnostics Network. What is your role? What's your region and what cropping systems dominate your region? Alicyn?
I'm the Executive Director of NPDN and also the Director of the Northeast Plant Diagnostic Network.
Mhm.
The number one crop in my state is potatoes, but then we also have 89% of our total land is forested land.
Sure. How about you, Peng?
I am a NPDN member. I used to serve as vice chair and chair for the accreditation committee, and I belong to the North Central Network. In Missouri, soybean is a top crop in the state, followed by corn and other field crops, as well as vegetables and ornamentals.
Okay.
So I am a member of the Southern Plant Diagnostic Network, and I have been part of the chair and part of the National Data Committee for a while, and I've been part of NPDN for many years, giving workshops and whatnot, and just being a member lab. And North Carolina is super diverse in the types of crops. In fact, most years we get over 500 types of plants and things like that coming in. But we have some interesting ones like Christmas trees, sweet potatoes, and tobacco that may not be found in other states as commonly, but we grow a variety of types of crops.
You're kind of unique in this trio because you're an entomologist.
The network is obviously fairly dominated by plant pathologists. I'm fairly unique in that I'm an entomologist and a director of one of these labs, but we do have several full-time plant pathologists in the lab as well. So I'm not the one diagnosing diseases.
Well, I mean, yes, it's great we have plant pathologists, but also we have to recognize that plant health involves both pathogens and pests. And I'm actually a little curious, like across the national network, how well represented are entomologists? Because when we think about vectors of disease, it's really important to understand insect and other arthropod vectors.
Yeah, definitely. There's a handful of us around that are part of the network, either in labs, in these central labs, or in the universities associated with the labs that kind of sign on to help with the diagnostics. Some universities have a separate insect diagnostic lab and disease diagnostic lab, but here our lab has always been a combined one for more than 50 years or so.
Alicyn, is yours a combined pest and pathogen lab or?
No, it's not. It is separated. But another thing that I was thinking about are nematologists. We have nematologists within NPDN too, kind of similar to entomologists. We have less of them than plant pathologists, but they certainly represent the members as well.
Sure. But I would definitely say that nematologists are probably more okay with wearing a plant pathologist hat than an entomologist is because, nematology is a subfield of plant pathology, a very important one, obviously, but certainly a lot of nematologists get a plant pathology education.
I'll also add, at least in the Northeast, we have a lot of diagnosticians that are entomologist trained, but carry out a lot of plant pathology. So, yeah, we have a wide array of individuals.
Yeah. And Peng, how about you? Is it a combined lab? Do you have an insect diagnostics lab there as well?
My lab was advertised as providing plant disease diagnosis, insect identification, and weed ID. That's why everywhere I go, when I give a talk or give a training, I told everyone that I work as a group. I'm not work by myself. Every time I receive some samples, I will do my own screening work. No matter it's a weed ID or mites ID, insect ID, and a plant pathogen ID, I do my best. And going further, I will ask my team member, like entomologists or urban entomologists or field crop entomologists or weed scientists, so that we will better help the clients to address their needs.
Yeah. I like the fact that you brought up weed science because that's obviously an important component of these things because we're dealing with all kinds of weeds that are supplanting native vegetation and impacting agriculture. So just coming back to this, role of vectors, I suspect that Peng and Alicyn, your insect vector identification is pretty well established.
I think part of being a good diagnostician is knowing your limits and knowing when you need to tap on someone else. And then also kind of like piggybacking on what you're saying, I feel like over the last growing season, we've had really interesting weather patterns the last 5 years. And so especially in our trees, we're seeing a lot of stress. And so you also have to have a good memory about, oh, we had a drought 3 years ago and that this tree may have been predisposed. And so now we have all these pathogens that are considered weak, but acting like big tough guys on these trees or insects. And so, yeah, they definitely stack the deck and it makes it incredibly challenging to diagnose tree issues right now in our state, at least.
Yeah, that's a great point. I like this idea of like having a memory for things. There has to be that continuity. Okay, so I think it's really important for our listeners to understand it's just how many samples you all process and just trying to understand the sheer volume of materials that like pass through, across your benches and through your labs. Maybe you could tell us a little bit about the quantity of samples or the types of samples that come through your clinics and how you go about processing them. Alicyn, you want to start with that?
Yeah. So it depends really on the weather. Last year we had a really bad drought, so we got less samples than normal, but I would say our average is about 1,000. So remember that this is just plant diseases coming into this lab, but we do a fair amount of research in this lab as well, so I don't count any kind of research effort samples. But yeah, we get physical samples, and then we also get digital samples too, so you can upload photos to my website for a small fee, and we'll diagnose that way. So we get them in those two forms.
And I suspect there are people that submit soil samples to soil testing lab. You probably have a separate soil testing facility on campus as well.
We do, yeah. Yep. But we also get soil samples to test for things like verticillium, and even we dabble in nematode testing. We might expand on that. I think as the director, you get to decide what interests you the most, and then you get to decide if you want to go into that area. And I have people that are interested, and I'm like, no. But yeah, so we get soil samples as well, but more for like organisms.
Okay. Peng?
Before I took this position, my lab has been closed for almost 2 years.
Mhm.
First year I started my position, I only received 214 samples. I was like, I love this job. I'm spending 365 days just working on 200 samples. And then the sample number started climbing as I got the name of the lab out, spreading the word. And now in average is 600 to 900 sample per year, including both digital sample and the physical samples. Half of those samples are field crops. The other half will be ornamental, vegetable, and other requests. Sometimes the number may vary, depends on whether my lab is involved in any survey samples or research projects.
Mm. And how about you, Matt?
Yeah, so our lab gets close to 3,000 samples a year, and I probably identify another several hundred email photos that are just sent to me that are, just randomly sent, or phone texts or things like that. And we are fortunate to have 3 full-time pathologists on staff, and they kind of divide up the different crops, and then the home ornamentals are kind of split up between them all. So we have one that focuses on vegetables, one that focuses on commercial ornamentals, one that focuses on field crops. So I'd say each of us look at about 500 or 600 samples each year. We also do have a separate turf diagnostic lab that is just for pathology. I do get the turf critters that come through, but they're a separate physical lab, but they use our same system, communicate all that stuff through the same system that we use. And so they have pathologists there that work just on turf internationally. Actually, they have an international permit to receive from countries all over the world. So that's kind of generally what we do. And we get a variety. We do get image samples and physical samples as well. So
Yeah. Turf was probably a big industry down there, or at least the golf course management is a big industry down there, correct?
it is, and this is they're pretty well recognized turf diagnostic lab for pathogens, and of course, yeah, the Southeast, especially Florida areas like that, have really big turf industries.
Yeah. So I mean, when I add up just coarsely across the three of you, 5,000 samples across your three institutions, excluding the other labs that process soil and turf and those things. Alicyn, do you have a sense of overall across the 70 cooperating institutions that make up the NPDN, how many samples do you think that are seen across that entire network?
One piece to tell you the breadth of samples that we get is that it's like 98% of all counties in the United States diagnostic labs have received samples from. So thinking about the distribution of where samples come from, it is pretty staggering.
I'm looking here, and total since I think 2001, I think that's about when they've been creeping records, 2.8 million samples.
That's impressive. And it just speaks to the importance of the network. I mean, the knowledge gained across those samples and plant types and the ability to lean on other members of that network to say, you know what, our region really hasn't been a big player in this industry or this crop, but things are changing and And all of a sudden we're seeing these problems. And then, Alicyn's scratching her head and Peng's like, oh yeah, I've seen that. That's very common for us here. So it's great that we can kind of lean on these different regions and these different members.
Yeah, definitely. I mean, I think it was last year, Matt, I sent you images of a mite, right? I mean, we just doesn't matter what region you're in, your expertise, you just tap on the people that you need in the moment to make a good diagnosis.
Yeah,
Yeah,
we all love doing this and helping people, and those people could be other members of the network. And I have a whole network of expert entomologists where if I don't know what it is or I just want to confirm something, I'll send some photos to folks, and yeah, it's really helpful to have that outside expertise.
Yeah, I agree. So, I mean, 5 years into my current position as a lab director, I didn't sense any kind of like a competition from other labs. We are all like big family. We help each other. And there are even some clients received a sample from Missouri. And there's some lab received a sample from Missouri. They actually call me and ask me to connect with the client for further questions. So that's pretty neat.
absolutely. Matt, I really appreciate you saying that, like, you're like, this excludes the emails and the texts. And I, to this day, get random texts of plant diseases, and I have no idea who the person is, and I'm not going to admit that I don't know. And I'll just give them an answer, and I'll just be like, I'm doing free diagnostics for strangers. Alicyn, Peng, is this something that happens to you as well?
Yeah. Yeah, I'm not gonna lie. There's been a moment when I went to a yoga class and people were talking about plant diseases, and I just needed to not think about plants in that moment. And I just did not say a word and pretended to just look down because everyone will talk to you about their plants and all the things. So
Yeah.
There for us, we beginning last year, we established an online digital submission system. So, I mean, previously, some people just text me or send me two photos without any background, without any kind of like information. They were just like what it is. And I need to spend more time to communicate with him for further information. But now with this system, you can scan the QR code and log into the system and upload a photo, add some information about history, about the plant information, disease history. That actually help, even though not all people like that, but that actually improve the whole process.
Yeah, absolutely. Diagnosticians are usually generalists that have training in a lot of areas, but there's still this fact that there's a lot of plants, for example, that I've never seen, you've never seen, and dealing with ornamentals, you're gonna have stuff come across your desk that you have no idea what it is. And if you're in like area like Florida, Carrie was talking about this, where you have like tropical and subtropical plants, it's even more complicated. How do you approach something when you get a non-agronomic crop or a non-native plant that you're trying to figure out what it is and then what's wrong with it? Alicyn, you want to start that off?
yeah, so yes, I mean that certainly happened to me before, and Pong and I actually were in the Florida lab for 4 years, so we know that feeling of like getting a lot of plant samples that we're not used to. And kind of what I teach is like that's kind of step one, like identifying what the host plant should be looking like, so that you can determine the symptoms it's expressing that
Yeah.
it shouldn't be. Like, what's a healthy plant? What's an unhealthy plant? That's where you start. And so, yeah, I have suggested to students in my lab and, even when I teach, like, yeah, use your apps, use your books, use Google, use Bugwood, use all the resources out there, trustworthy sources, because they're at your disposal. And that's definitely where you want to begin.
Absolutely. Pung, do you have any other things to add there?
I use the app called Picture This.
Uh-huh.
So kind of like a pre-screen to give me an idea of what the planet is. I'm grateful that those weird tropical plant normally submitted by some people they know the plant very well, such as Missouri Botanic Garden. He had a lot of exotic plants and they know well, but they just don't know about disease. For me, as long as I know what the plant we're dealing with, I just study the plants and check all the common disease associated with that, and also consider all the factors in their own environment condition. But every time I do all the plant ID, again, this is just like pre-screening test. I always give to my weed scientist who is a professional for plant ID
Yeah.
for second opinion.
Yeah. Matt, I'm thinking about you and your expertise. And oftentimes when I teach the second half of my forest pest management class, which is dealing with the forest entomology side of things, I talk about the importance, just like on the plant pathology side, of knowing the host. Like if you find what looks like a herbicide burned locust tree, late in the summer, you're like, well, that's a locust leaf miner. Like just knowing the plant host obviously helps inform your diagnostics. Do you get a lot of insects like on plant material, or are they more like just arriving as insects decoupled from the plant tissues?
Yeah, that's a great question. And the funny thing about me, I guess, is that my background is more in medical veterinary entomology and in systematics and taxonomy. But luckily, I do enjoy plant ID and plant taxonomy. And, I really enjoy wild hosts and ornamentals and things like that. So I like to dabble, and I've definitely learned a lot while being in the lab as far as identification. We still have questionable samples that either come in or we have photos of them. But we also have some other people in the lab that are experienced with plant ID, have seen a lot of plants. We also know if we can't ID a plant, oftentimes that plant is probably never gets sick or pests because it never comes to the lab. And then finally, we do have very good botanists in our university that we will consult either in the crop science department where they're weed specialists, from aquatic to pasture weeds. And then also we have a botanist that works in the herbarium that is really good at identifying kind of wild plants and a lot of the woodies and things like that out there. So we just rely on experts if we don't know. But even though we get a huge variety of plants, we get most of them we are familiar with.
Yeah. So I want to get into an interesting question. What do you think are like the biggest misconceptions about plant disease and plant pest diagnostics? Alicyn?
I would say that every disease that we identify should be managed. I feel like that's a misconception. Sometimes it's best to just walk away and not look so closely, right?
I talk to my class about this all the time. Is I can give you management strategies, but if the cost of managing exceeds the profits you'll make from harvesting the crop, then you're not going to implement that management strategy because you actually can't afford the management.
Right.
You know?
Yeah. It might be even cheaper to replace. I mean, depending on the cropping system and what kind of plant you're talking about. But yes, or just being smart about it. So if you know what you're dealing with, then kind of being strategic about just kind of living with it, whatever that disease is too. I sometimes take that approach with individuals or give them a number of options of like how they might be able to manage it. Because everyone's, especially for homeowners, like your connection with a plant plays into the importance of that plant. And
That's
the
right.
value of that is changed if it was passed down or the person that gave it to you is no longer alive or, all those things. So you have to take all that into consideration when crafting a management plan for someone.
Absolutely. How about Peng? How about you? What do you think is, we can come back and do a second round of this, but I want to get everybody's opinion first here.
I think I have like 3 misconceptions.
Okay.
First one is all diseases are caused by either pathogen or pest. So I have a client who are not happy with diagnostic report saying that no pathogen found, environmental stress suspected. They're not happy. I don't know. They desire pathogen even more than a plant pathologist. So, and Missouri, the climate has been swinging very dramatically over the past few years. We have a lot of environmental stress issue in Missouri. And most of the time when I receive a sample like arborvitae, boxwood, or white pine, they're environmental stress related. No pathogen found. So this is something I have been repeatedly telling a client that there's nothing we can do. I have already ruled out everything. So this is no pathogen found. So that's the first thing. The second thing is the homeowner or the grower in Missouri, I always let them know that you are unlikely get an invasive species or pathogen or pest from the headlines of national news. So they always, when they have some problem with their plant, they immediately thought about some disease they read from news, like a boxwood dieback. Even landscaping company or lawn care company, they immediately draw the conclusion about those bad, notorious disease.
Yeah.
So that's a second misconception.
Yeah, yeah. Well, I want to talk about that a little bit
Yeah,
because I feel like it's analogous to WebMD. It's like you're like I have left ear pain and my knees throbbing, and you're like in 1 out of 7 billion cases it could be a rare cancer, and you're like that's what it is. You know, you always go to the most extreme thing because you're like it has to be that. So it's a little bit analogous in that way. What's your third?
the last one I second Alicyn that people always think about spraying chemical and even spray chemical before sending sample in. They tried all method without even ID the problem. And they always, once they receive report, they saw a really mild leaf disease. They immediately think about what should I buy? What should I spray? But in most of the cases, you don't have to.
Yeah, Matt.
I would say the biggest misconception is that we know everything and don't have to look up things after a couple of years of not seeing something. And, it is definitely beneficial to have a good memory and good, lots of experience. It's really difficult when you first start out and you everything is new and then you kind of file away the things that you have experience with. But there are definitely things that are always coming in that we don't know or not like instantly. If somebody brings you something and they're like, why don't you know what this is? It's like, well, there's tens of thousands of arthropods in North Carolina and stuff. And so I don't know every single one, but there are difficult situations where people want to just kill everything, even if it's not a pest, or people that just don't like critters that aren't like them in general and want to kill everything, or want to be completely sterile, have a completely sterile environment and every plant be perfectly healthy and no chew marks out of them, nothing. And I think they don't understand the fact that a lot of these insects that we like and whatnot come from places that you don't think about.
Yeah.
Like I like talking to people about where, have you ever seen a baby firefly? Nobody even thinks about what baby fireflies do or where they live or what they look like. And it's just like, oh, fireflies are just there and that's it. People take things for granted, or they just assume things are all pests. And of course, for entomologists, one of the biggest issues is delusional infestations, where people are convinced that they're infested there. And so that is a very difficult part of our job as entomologists, I would say.
Yeah.
And I would say I've seen in the lab that there's some things on the plant pathology side, like, my neighbor infected my plants with fungi and like some nefarious thing that they think or whatnot.
Yeah, I like to come back to this, like, we have all the answers, we have all the solutions. I talked to Carrie about this too. It's like we may not have a solution, and also we can't perform miracles. Do you have anything to add, Alicyn?
Just that I agree with all of it. Yeah.
Yeah. Yeah, well, the thing that Peng said about all diseases are caused by biotic agents, I think that's something I teach in class that, there are nutrient deficiencies and there's air pollution. There's, things like hail damage, frost damage, and these may open up the plants for opportunistic invaders. And they may be the ones that are, determined to be the causal agent, but they were predisposed at first by this, by this hail damage or by this nutrient deficiency. And, leaning on these other things like the soil testing lab and these things could really offer a more comprehensive snapshot of what's happening in your garden or on your land. So, staying on that topic about abiotic causes of disease is like Alicyn, of the plants that, I think you mentioned about 1,000. How many of the plants you receive are you seeing more related to abiotic disorders and things like that versus biotic agents? And is it more that you're just saying no pathogen found and it's probably that?
Well, I would say maybe the more interesting answer to that is this growing season for me, I felt was the most challenging. Like I had the most unidentified diagnoses that I've ever had this growing season. And I honestly, I think it's because of the environment, like I said, but also because we have the technology now to definitively say, no, it's not these things. So there were a number of, and they ranged from like lettuce to trees, where I went through everything and like scratched off the list, all the possibilities. And then I just decided, because I wanted to know, we sequenced it just to see what's in there. Like, what may I have missed? And then it came back as nothing. And so I feel like, a diagnostician 20 years ago, they would have said suspect and then said what they thought. Whereas now we can actually say like, oh no, there's really nothing there.
Yeah. Actually, what you brought up, the technology to move forward and actually get to an answer, brings up another important point that I forgot Carrie had brought up. And it's about how much you should and how much we're willing to spend on a single plant or a single diagnostic, right? Like we have all these assays we can do. And I don't know, I want your perspective of, do you charge and is it a fixed price? And if you don't charge, like there's got to be a limit to the amount of tests you can run. And certainly your diagnostic skills allow you to process of elimination to say, it's not this, it's not this. So it could be 1, 2, or 3, and then you perform some molecular assay. Do you want to speak to that a little bit, Alicyn? And then we'll hear from Peng and then Matt.
So oftentimes we'll put that on the submitter, like how much are they willing to pay? So about a month ago, I had a maple tree that had a bleeding canker. The forest pathologist and I couldn't figure it out. They really wanted to know what it was. So they paid $250 for us to sequence it. We got the results back. We know what it is now.
Your point was that, if you're willing to spend $250, you'll take it and run the extra tests.
Yes.
But know that it doesn't guarantee you're going to get an answer. It's just that you're going to eliminate all the things you thought it could be.
And that we were at a dead end. And this is another possibility, right? It's like human diagnostics. Like these are all the things that we've done. There's only this one final thing that we could do. Do you want to do it? It's this amount of money. And then they say yes or they say no. But then also, and I know all these individuals, like us as diagnosticians, these stick with me. I mean, I remember ones from 7 years ago that I never figured out, and I'm still trying to work those out. So like, I'm also willing sometimes to put the extra effort into it to figure it out because I personally want to know and that it will help me in the future make a similar diagnosis. And so I am, this year, I was sequencing a lot of stuff because I was at a dead end. And, we're also doing some really cool things too. Like we've had someone who their pipes burst because it was filled with tree roots. And they didn't know because of their landscape what tree it was that they needed to take down. So we sequenced the roots. Spoiler alert, it was the apple tree. Who would have thought?
Yeah.
So we're doing really cool things to like help people get answers, even if it's out of the plant pathology realm.
Yeah. Okay. Let's move on to Peng there. And Peng, what in terms of some of that discussion about technology, the cost of diagnostics, do you want to speak to that and your interactions with clients on this?
We have the similar approach with Alicyn's lab, and we have the base charge, which is only $15 for every client. And if there's any further tests are required, I will give a phone call to the client to see whether they agree with it, whether they want to move forward. But I do have exception for a home gardener, a home grower. Their bill will not be on $30. So like $30 is like a limit. I won't charge them too much. But for crop consultants or ag companies, I will just build it up. Like ELISA test, $20, PCR, another $20.
Yeah.
Sequencing another. So that can go all the way to like $85 to $100. So I do have kind of like different groups that are charged differently.
Sure. You have to kind of speak with the industry folks and the bigger companies. They know they've done this before. They know what they're doing. You get a home gardener, they might come in with pretty high expectations. Do you have to kind of like help reset the expectations for a lot of these like first time users who think you're gonna like not only perform miracles in a timely fashion, but actually, solve all their problems?
Yeah, it's really hard to satisfy all the home gardeners. I would just do my best. Like a tomato disease problem, I examine the leaves, the roots, and stem. I'd also run a couple ELISA, try to rule it out, all the potential pathogens. And in the end, if there's no pathogen found, the plant is still dying, I would encourage them to replant as soon as possible, such as sometimes herbicide damage. User cannot do anything with that, but my lab cannot test for herbicide residue. For the home gardener, I just let them to regrow and replant as soon as possible.
But you can look for symptoms for, herbicide damage.
Yes.
Because there are obviously some unique symptoms associated with certain classes of herbicides. So you can get a little bit of insight in that regard. Matt, how about you? What's the charge there? And what if people want to find out a little bit more?
Yeah, so we offer free image sample diagnosis, but for disease and many things, that's just kind of the first step. And then we often have to request a physical sample. For physical samples, we do have a flat fee. We offer a reduced fee of $20 through the extension service. So that way they can bring it there and maybe they can answer that for free in the county and we don't ever have to see the sample. But they also, the county extension office can ask the better questions, can fill out the form better, and they submit it in a more proper way. And so we do get that added benefit. So we encourage people to go for that. All other direct submissions, except for out-of-state samples, are $30. And it's more of we do all our accounting, all this other stuff. And so a la carte kind of stuff becomes a little bit complex when it comes to billing and things like that. So we've kind of have just a standard rate. But yeah, it's just like, everyone else said that, that's actually the value of a diagnostician is AI can't really, or, robots can't really tell exactly which one's the first thing to do. We know, okay, what's the most likely, we're going to try that tactic first and then keep going. And then at some point we need to say, is the diagnosis worth the amount of extra effort, which, you may strike out with. So I think just understanding what the process is and how complicated it could be and what kind of answers you can give the clients is really important.
Yeah, and I think Carrie said this best. It's like this idea where when you're trying to diagnose, if you see hoof prints, you think horses, not zebras. But it's really important that you know all the horses that are in your type of horses that are in your area. And I think that's really good. One of the things I want to talk to, and I think Alicyn, given you're the director of the NPDN, you might be offered the best insight here is like, say we have a quarantine pathogen show up in a sample submitted. Of course, when that comes into play, there's a lot more, the due diligence you have to do is increased and additional testing. Does the lab or the network take on the additional costs of paying for additional diagnostics when a quarantine pathogen or pest show up in a homeowner sample or a submitted sample?
So if I was to diagnose something that's quarantinable, there are certainly a number of steps that need to be taken. The first being contacting your state SPRO, and then they will basically tell you what steps need to be taken. And oftentimes it's them going out and surveying, that's like the proper procedure, and then them sending samples to you. So it really, I guess financially, it is handled in a way of like a normal sample.
Yeah.
There's no real difference there. There are some pathogens that there are funds dedicated through APHIS for kind of passing on to diagnostic labs for testing. That's not the case for all of them.
But would you, in any instance, I mean, at the same time you're contacting your state offices and getting some feedback from them, do you request a second sample from the homeowner because you want to confirm it and you could absorb that cost? Is that something typically you do in a situation where you find something new, maybe not even quarantine, like the first, I mean, it doesn't have to be like the extreme case, but maybe it's something new you hadn't seen before. Is it common to go back to that landowner or submitter and say, we'd love to receive some more samples just to further look at it?
Yeah, I've definitely done that. And we all just kind of filled out a survey indicating our first in states that we found. And, it's pretty common. I think I had 48 first in states in 2025. So, some of them more interesting than others, obviously, but I definitely didn't ask for a resubmission on all of them because generally they'll send pretty good samples and we ask for a little bit more tissue than we're actually going to use. So if we keep it in the fridge, we can go back and dip into that if we need to. One thing that came to mind when you asked that question, some states, such as mine, serve as the Department of Ag diagnostic lab. So some Department of Ags have their own diagnostician, but some don't. So I work very closely with my Department of Ag because of that.
Yeah. And I think about, as a tree pathologist, I think about these tree care companies like Bartlett and the bigger ones, and they too have their own diagnostic labs. Like Matt, is the Bartlett's in North Carolina or something like that?
Yeah, the diagnostic center is in Charlotte, North Carolina, and they are part of the network. Actually, they're a really interesting group because they're a private company, but
Yeah.
they're very academic. They really enjoy the diagnostics. A lot of the people kind of are hired even from our networks and our labs and things like that. So it's a really interesting lab, and they do a ton of samples each year. They do a lot of samples.
Yeah. What's the craziest thing you've ever seen or dealt with in the clinic that was unexpected or just that is memorable?
The strangest samples, yeah, there are some weird ones out there. I did have a client who was afraid that cicadas that were landing on their house were government spy devices, which was fun to try and convince them that they weren't. And yeah, I wrote up a long report about that just to be thorough.
Yeah.
The weird kind of like one of the things I talk to people about is we have this one specimen in our collection. It's the only, so far as I know, the single specimen we have for this species in our insect collection, and it was collected in a child's hair, and the mother thought it was a flea and things like that. So we wouldn't have had that specimen if it wasn't for the child's hair catching it. And yeah, there have been some interesting situations or like pests that emerge, that are kind of explode every 10 years, and then you never see them, so nobody knows what they are, and they said to me, like, oh, that's interesting, and then actually help trying to figure out with the specialists, the extension specialists, how to manage these things that have not been seen or haven't been an issue in 50 years is, sometimes they ask me about it, and I have to say, I'm the diagnostic expert. I don't do a lot of management. And in fact, that's sort of when I talk to people, I tell you I'm probably the most annoying person you'll meet because I can tell you exactly what something is, but I can't tell you how to kill it, except for in ethanol or in a freezer, which isn't very viable for widespread management. So yeah, but we get some strange insects. I get some specimens that are either new to science or new to the U.S. or the state, and just ones that are really interesting and new to me. And I did want to follow up on what Alicyn said. We do ask for follow-ups a lot, samples, and we don't charge for that because we want to encourage people to do that, especially regulatory samples. And in fact, if there's a sample that we think the person might not submit on their own, but it would either help us for our education or would be really important that we think it's new or something important, we will waive the fee on those to encourage them to send it in, because we know that we're asking sometimes a lot of people to go back out to a site and collect it or whatever. But we've had many samples that have come in that we really need to see in person.
How about you, Alicyn? What's the craziest thing you've seen come through?
So my office wall is shared with the veterinarian diagnostic lab director.
Mhm.
And so she does necropsies. And this growing season, she gave me what she found in the stomach of an alpaca to help her identify if there were poisonous nuts that were the cause of the death. I was looking at that under the microscope thinking, who would have thought? Who would have thought I'd be here? But it helped her. It helped her make a diagnosis. And that's totally something I enjoy. So, yeah.
And how high up on your CV is that?
Well, yeah.
Skill?
Well, great point. I need to add that to the top. Thanks.
Yeah.
Alicyn, you've reminded me of a couple other things that you could say afterward. If yeah.
Well, I can I just add another, which isn't like a diagnosis or anything like that, but I just have to say it because it happened in Carrie's lab and she has no idea. So I just think this will be funny and we'll definitely figure out if she listens to this or not. So I worked in Carrie's lab for 4 years and I was in the BSL-3 section of her lab. And, as you always do, you clean the benches with ethanol. And then I went to go, I wiped it down. And then I went to go light the burner and the entire counter lit on fire.
Yeah.
And I was like, oh, okay, yeah. But it was real clean. And the person standing next to me, we decided just like, let's never talk about this ever again. But now when I train my students, I do tell them, be careful of this.
Yeah.
So.
It's fun.
Wait, as an entomologist, you're not supposed to do that? Oh.
How about you, Peng?
I feel like the entomology world is more crazier than the plant pathology world. I actually had one case similar like delusory parasitosis that I received Ziploc bag with dead skins of a client she collected for 3 years. It's a big bag with white powder, and she asked me to go over it and look under microscope to check the insects. 3 years of dead skin collected from beddings, couch, everywhere. So I would consider that's a crazy. But I also have a fun crazy example is I have a client who was about to spray fungicide in the afternoon. He gave me a call, asked me what I think about it. I think that's a wheat virus. He doesn't need to spray a fungicide. And I said I need to do the ELISA test for his wheat. And he flew his airplane to Colombia and drop off the sample in just 30 minutes.
I've really appreciated talking with all three of you at the same time.
Yeah, thanks, Matt. Thanks for having us, and great to see you, Alicyn and Peng.
Yeah. Thanks.
Thank you. Thank you, everyone. Thank you, Matt, for the invitation.
You've just heard from Dr. Carrie Harmon from the University of Florida, Dr. Alicyn Smart from the University of Maine, Dr. Peng Tian from the University of Missouri, and Dr. Matt Bertone from NC State University. Be sure to check out our show notes for links to learn more about each of our guests today. I'm Matt Kasson, host of Plantopia. Thanks for listening.